Dear audience, the next speaker for the day is Dr. Sudhir Sarangi. He has completed his MBBS from MCCT Medical College, Berhampur, and then pursued both his MD Pharmacology and DM in Clinical Pharmacology from AIIMS, Delhi. Sir has areas of research interest in clinical pharmacology, neuropharmacology, pharmacovigilance, drug safety, pharmacoeconomics, and pharmacogenomics. He also has several publications in reputed journals like Indian Journal of Pharmacology, Psychopharmacology, European Journal of Clinical Pharmacology, Saudi Pharmaceutical Journal, to name a few. Dr. Sudhir is also the deputy coordinator of the Adverse Drug Reaction Monitoring Center at AIIMS Delhi under the Pharmacovigilance Program of India. He has authored a total of 8 chapters in popular books, namely Advanced Pharmacology, IAN Textbook of Neurology, and Nanogels in Biomedical Applications under the Royal Society of Chemistry, United Kingdoms. Sir has been bestowed with numerous awards and honors, like granted membership by the National Academy of Medical Sciences, obtained Certificate of Achievement for poster presentation at the 34th, 33rd, and 32nd International Epilepsy Congress. He was also awarded the Best Paper Award in the 20th Joint Annual Conference of the Indian Epilepsy Society and Indian Epilepsy Society Econ 2019, among many others. Today, Sir will be speaking on the topic of collaborative research networks. So kindly welcome Dr. Sudhir Chandra Sarangi, and over to you, sir.
Very good afternoon to all of you. Let me share my screen. Okay. So today we'll deal with collaborative research networks, but basically we'll discuss with in line with multi-centric clinical trials. And we'll also discuss some points and some issues relevant to the multi-centric clinical trials. As you know that clinical trials is any research which involves human participants, and it can be a clinical study, means it can be clinical trial or observational studies. So clinical trials evaluates the effect of certain interventions and its effect upon the health outcomes in a healthy participants or in a patient populations. So if you tell about the multi-center clinical trials, that means a clinical trial being conducted at multiple sites or multiple centers at the same time. And global clinical trial means any clinical trial which is conducted as part of a multinational clinical development. That means same clinical trial is being conducted at multiple countries. It is called multinational clinical trials or global clinical trials. If you'll see the current picture of how many clinical trials are being conducted as multicentric, I means as per the discussion by Dr. Prasad, all the clinical trials are to be registered with the clinical trial registry. So here I have taken the EU clinical trial register, clinicaltrial.gov of USA, and clinical trial the clinical CTRI of India. If you see this phase 1, phase 2, phase 3, phase 4 trials, see here, as per the EU clinical trial registry, till March 2023, 347 clinical trials has been registered, and in same clinicaltrial.gov there are 1764 clinical trials are registered. And this column is showing the clinical trials that are registered in clinicaltrial.gov sites and where India is a site. Okay. So coming to phase 2, similarly, these are the study numbers. And so phase 3 are the majority of the studies are phase 3, which are conducted as a multinational studies or multicentric studies in all the trial registries. If you will search these multi-center clinical trials in CTRI, you will find around 1109 studies or records relevant to these multi-centric clinical trials in CTRI registry. That means number of clinical trials are being conducted as multi-centric trials. So we'll see what is its relevant and what is its issues. And this global picture is showing that as multi-centric clinical trials are not only limited to the developed countries, all over the world, different study sites. It may be a economically weaker sites or a developed sites. All the countries are participating in multicentric clinical trials. Why it is being so? to have the external validity of the clinical trial results. Okay. So coming to history, yes, the Patulin trial is the first multicentric clinical trials conducted in the world, and it is conducted in 1944. And this trial is about a chemical that is patulin, which is derived from the Penicillium patulum. It is a fungus, and it is tried for the common cold. Before this multicentric trials, several separate clinical trials, individual centric trials has been conducted. Case series reports has been reported. Some are having effective results and some are having no doubtful results. So based upon that, a group of investigators planned for these multicentric clinical trials, and these results have showed that this patulin has no efficacy in case of common cold. But this trial has shown the path that yes, there is need of a robust clinical trials platform to have a sound result of any study, instead of doing separate clinical trials and wasting so much resources. And based upon this, the next multicentric clinical trials was conducted in tuberculosis with streptomycin and paraamino-salicylic acid. And this is conducted in multiple sites. You can see 1, 2, 3, 4 like this, 8 to 10 centers in England. Okay. So coming to current study scenarios, we have faced the COVID pandemic, and several studies have publications have reported that what is the clinical trial failures or clinical trials lacunae in this COVID pandemic. If you see this clinical trial landscape, till June 3rd, 2022, more than 2,700 therapeutic trials and 1,752 vaccine trials has been conducted worldwide for COVID-19. But can you count how many currently active drugs are for this COVID-19? We have a very limited drugs. Even the remdesivir, which is considered as a brahmastra for this COVID-19, is now showing some side effect profile like bradycardia, and it is being doubtful to be included in the common treatment of the COVID pandemic. So more than 100 unique therapeutic agents has been investigated, but there are several duplicated trial efforts. And dose regimen comparisons are absent for multiple therapeutic agents. And one of the most common findings is that multiple redundant control groups are being considered. Same therapeutic agent is being considered for clinical trial in AIIMS, in NIMANS, or in GIPMOR in different study sites. Then control group will be same, where we are giving the standard of care. But we are considering multiple control groups because the trial is being conducted in multiple study sites. If we will consider a multi-centric trials, then there will be a single control group and it will have more robust results. So this COVID pandemic has shown that yes, we should have a robust clinical trials where there should be single data repository. There should be smarter investment or smarter fundings, means the funds should be utilized in a purposeful manner. As Dr. Sarik has discussed, that platform trials or adaptive trials are the best scenario for any good robust clinical trial results. And it is based upon multiple arms where one arm can be included, one arm can be dropped out based upon several interim evaluations. One more important thing is that there should be a master protocol. Yes, it is a single overarching protocol designed to answer multiple interventional questions that would require separate clinical trials. That means if you are planning to do separate clinical trials in different centers, it will be better to have a common protocol that is called as master protocol, and that should be followed in each centers. But how to do that, that is a question mark, and that is the issue also. So master protocol with platform trials, it is required for the large trial network, means good collaboration is needed between the investigators, the participants, the regulatory bodies, and it should have a common infrastructure across the investigational sites and with multiple institutions. Means the requirement of site should be similar across the different study sites and that are able to join and live over the time. Okay. So And one more thing is that there is need of efficient collaborative research ecosystems. Collaboration is a more important thing in case of multicentric trials. We'll discuss how this collaboration at what endpoints, at what time points it is needed, and in between whom it is needed. And national level collaboration and buy-in from major stakeholders means from how many funding agencies we can obtain the funding. If we are conducting a multicentric trials, then that will be a more good idea that we are getting more fund for a single trials. So reasons for multicentric trials, that means it is providing sufficient subjects, that means power of the study will increase. Generalization of the study results, that is external validity will be quite high. And diverse expertise thoroughly investigate the pitfalls in a study protocol and conclusions. Multiple investigators are there. They can opine themselves what is the better idea to promote this or to have a good protocol, then it will have a good results. So coming to the largest multi-center clinical trials as per the data till now, it is one of the trials conducted in India. Yes, it is the Delphi trials. The Delphi trial, it is deworming and enhanced vitamin A supplementations. It has been done over a period of 6 months supplementations, and it has enrolled around 1 million preschool children. Okay, this is the largest multi-center clinical trials till now. Coming to recent COVID status, we have recovery trials, we have solidarity trials, and in case of COVID recovery trials, you can see there are 48,465 participants enrolled till now, and it has been active in 195 study centers till now. Coming to India, the COVID vaccine trial is also a bigger and you can say larger multi-center clinical trials, which has enrolled 25,800 subjects across 25 sites in India. And this is the global trials as for the solidarity trials. There are 600 studies hospitals are involved, 52 countries involved, and 14,200 hospitalized patients were admitted or randomized in the studies. So multi-center clinical trials is also defined and regulated by certain guidelines. So this ICH E9 depicts how to conduct a multi-center clinical trials. So it should be at any stage of clinical development it can be employed. It should have a common protocol and standardized procedures. There should be investigator meetings, personal trainings, and careful monitoring to reduce the variation of trial conduct and evaluations. And a good design is needed, and combining the centers in the analysis. It is not that one center is not recruiting any subjects or it is giving totally negative results, so we should not include the study results from this side. No. All these study centers data should be included in the analysis. We'll see how this analysis is to be done in later on slides. So coming to issues, where the issues can be, yes, issues can be at the pre-planning phase. Pre-planning phase, it is not the planning phase also. Planning phase, ethics related to ethical approvals, conduct of the studies, what should be the sample size, how to have a sufficient number of sample size, how to do the randomizations, analysis of the data, how to do the proper analysis, whether treatment effect should be considered, whether the study center variability is to be considered or not, then how to do monitoring, how the regulatory inspection is done, and regarding the publication. So we'll deal one by one. So coming to pre-planning phase, yes, first of all, we should have an idea, that is research question. And the research question for a multi-center clinical trial should be finer one. Finer means it should be feasible, interesting, novel, ethical, and relevant one. And how to devise this research question? At least we should have certain background data. The background data can be based upon an external pilot study or an internal pilot study. External means it is not part of the MCCT. Means it is a separate study that has been conducted earlier, or you can conduct a separate small scale pilot study, and it provides the feasibility and error margins for the large multi-center clinical trials. Instead of that, we can conduct an internal pilot study where this internal pilot study data can be included in the final analysis of the MCCT. And coming to the planning phase, we should assure that a more generalizable sample of the study is being or is going to be enrolled. It is not that we are going to enroll the study subject from a good hospital where the sample size can be easily obtained. Or we should include only those study sites which are having good name and fame. No, it is not going to give a external validity of the study results. We should include the study sites all over the country where it is feasible and where the chances of variations are there, which can be described later on. It enables the investigators with similar interests and skills to work together on a common problem. But there are also disadvantages. What is this disadvantages? It can have more cost, complexity in planning, logistics, and variation across the centers. Some investigators may say that, no, I am not comfortable with this protocol plan, change it. So how to deal with it, we'll see it here. So coming to the planning phase, we should have a research team who should be highly functional, experienced, and role responsibility should be priorly ascribed. And there should be coordination and constitution of committees, like which committee should look after the data safety monitoring, which committee should look after the drug coordination committee or drug distribution committee, and who should be the statistician, who should be the epidemiologist in the studies. Okay. Next, collaborative centers. Yes, the centers should have extensive collaboration among themselves. And there should be feasibility assessment and training of the staffs. Coming to documents, the protocol, the study designs, a common data collection form, a common data reporting form, common ADR reporting form, common adverse event reporting form, all these should be there. And moreover, there should be manual operating procedures. It is for maintaining the consistency for data collection and adverse event reporting during the clinical trials. It is something separate from the protocol. Though all things are mentioned in the protocol, the MOP is just like a SOP to be followed at each study sites. Okay. So protocol is different thing and SOP is different thing. We will see what what is the difference between that. So MOP is just like a SOP to be followed at different study sites. Coming to protocol development, yes, it needs expertise, willingness among the members of the collaborating research teams, including epidemiologists and statisticians. And how to find out a perfect protocol? It should be by taking the opinions from all the investigators and the sponsors. But how to get a proper conclusion? So the proper conclusion can be obtained if we are considering all the investigators' opinions and giving them equal weightage. So it should be problem-oriented and face-to-face discussions with the intent of resolutions. Means we should conduct several meetings among all these stakeholders, but it should be intent of resolutions. It is not to intent of conflicting. So there should be a meeting coordinator or moderator should be assigned, and he should have a command attention and respect for all the collaborators, and a project lead should be identified. The project lead is a person who is dedicated to prevail through multiple recommendations and find out a solid decision or conclusion at the end. So what are the consensus building methods can be applied? We can have a focused group discussions or nominal group technique or Delphi methods. Nominal group technique means we'll ask for all the investigators or stakeholders to give their opinion how they want to modify these protocols. And later on, we can enlist all them, or the project lead will enlist all them in a flip chart or in an overhead projector, and it can display one by one point, which will be discussed later on among the stakeholders, and the final conclusion will be obtained. Or else we can obtain apply a Delphi method. So Delphi method is a multiple round conclusion deriving methods where a state of question is being employed to multiple stakeholders. They will give their opinion, and it will be analyzed by a unbiased or a separate or independent coordinators, and all the analysis data will be projected as a conclusion to all the investigators, and later on this round will continue and to find out a consensus among them. So coming to planning is the identification of clinical centers. As I mentioned earlier, that it is very important which study site should be a clinical center. It is not only the site having good name or reputations, it should give weightage to all the study centers to have a good external validity. We can appoint CROs, we can appoint or select the labs to which we have to provide these samples for analysis, and staff training should be equal at all study centers. You can see one of the examples, these recovery trials, how they have maintained the study site training and information for the site staffs. Yes, we can design such a web-based platforms where we can have a site setup, how to set up the sites. If you click this, it will give you detailed information. What are the regulatory documents needed to be kept? What are the data monitoring committee correspondence to be done? How to do it? How the safety data is to be collected, or adverse event monitoring data is to be collected? How the annual report is to be prepared? And if you click each point will give you two different confirmatory or different procedures to be followed at all study centers. So how to the pharmacy or the drug to be maintained, how the inventory is to be prepared, how to maintain the accountability of this drug or investigational products, what the site teams will do, how to perform training of the study personnel or the study investigators or co-investigators. Okay, how to do the randomizations. All these things are to be classified as a training manual to all study centers. So coming to coordinating center is among the different study centers, one center is identified as a coordinating center, other are the collaborative centers or study sites. So the coordinating center has very important role because it is to maintain the day-to-day trial activities of all the study centers, ensure IRB approval obtained by each study center, implementing the randomization scheme, collecting, monitoring, editing, analyzing the data, maintaining the documentation of adverse event report. Yes, it is very important. Suppose one SAE has been reported by a study sites. Immediately it has to be informed to these coordinating centers, and coordinating center or the principal investigator or the project lead should disseminate the information to all the study or all other study sites. Otherwise, more of the number of subjects will be subjected to that serious adverse events, and accordingly the cautions is to be taken. So it should facilitate the audits by regulatory authority. Means the regulatory inspection team can visit to all of the study centers or some randomly picked study centers, or they can come only to the study coordinating centers where the data will be collected from all the study centers, and all this data will be subjected to the inspection at this site only. So the thing is that a single center malfunctioning can be tolerated, whereas the coordinating center malfunctioning cannot be tolerated. Coming to project lead, he is the important person to maintain the coordination among all the study stakeholders and investigators. He has to submit this, get approval and amendment of the protocols. Likewise, one of the friends just called me that whether we have to get the endorsement certificate from all the study centers or sites while submitting a multi-centric study to ICMR grant or not. Yes, it is needed. You have to get the endorsement and investigator signature from all the study sites, and you can compile it as a single PDF file and you can submit it to the ICMR grant for funding. Okay, so protocol single and correct person at all study centers should be employed, and the thing is that it should not be rewritten or modified by any of the particular study site investigators apart from the project lead. And if project lead is amending it, then immediately he should get the ethical approval and inform to all other study sites. Then timely review of adverse events and review of timely submission of the data for study analysis. This is the important role of the project lead. Coming to protocol, yes, it should have a title page with name and address of each centers, coordinating centers, and the contact of the investigators. Then procedure for central registration of the patient should be mentioned. The common data collection form or data collection, how it is to be done, it should be mentioned in the master protocol. And similarly, the reporting of adverse events and the drug procurement plan and drug distribution plan. So all these things should be mentioned in the protocol. So I'm not discussing each point what should be there in the protocol. I'm just highlighting that what is the issues that can be faced and how it can be handled. See, regarding the recovery trials, same example. See, this trial is the trial which is being funded by so many funding agencies. It is funded by the Oxford University, by Wellcome Trust, Bill Melinda Gates Foundations, UK Research and Innovation Organizations, MHRA. So getting funding from so many agencies gives a bulk of funding or more funding which can be utilized for better study proposals. And this trial is not only registered at the single study register, it is registered at EU registers, the clinicaltrial.gov, and also in India also, CTRI also, because this recovery trial is started in India since 2022, June 2022, the first patient has been recruited. And one of the points you can see in this protocol, it has mentioned that in UK, longer term follow-up or up to 10 years of follow-up will be sought through linkage to the electronic healthcare records and medical databases, which is already maintained by the NHS digital or public health agencies. But coming across the status of India or some poor developed countries where there is no electronic health record is available, what should be done? Or we should not follow this point in at all in these protocols? No. Accordingly, you have to revise the protocol and you have to amend and mention this point in such a manner that it should be applicable to all study sites. What they have done, they have mentioned that outside the UK, due to the absence of electronic health data linkage, additional follow-up will be conducted at 6 months after the first randomizations by telephone or in person in order to collect information on mortality and readmission to hospitals. Means you should consider all the study sites and how this study proposal can be applied at each centers. Accordingly, you have mentioned all the points in the protocols. Come to the next example. In the same trial, it has been mentioned that in Indian context, if the patient regains if the unconscious patient is to be enrolled in this study, then the LAR or legally accepted representative can give the consent. But whenever the patient regains consciousness, you have to take the ethical approval from that subject only. But as for the recovery trials, you have to adhere to all study sites regulatory documents. So as per the point mentioned here, highlighted here, if they regain capacity, such participants should be provided with the information about the trial, what their rights and how to exercise them. But it is not necessary to obtain their written consent. And the asterisk C is there. And it is mentioned that unless required by the local regulations, this is not required in the UK. So that means you have to adhere to all the regulatory requirements of the individual study sites. Accordingly, you have to mention all the points in the protocol. Okay. So coming to ethical issues, yes, it is one of the point that it is unethical to include patients from only developing countries or underdeveloped countries for just sake of the cost and cost-cutting exercise. Previously, India is considered as the guinea pig of clinical trials, but somehow it is being regulated. The regulator has been strict now. So trials should comply to the local regulations. Consent processes should be in respective local languages. Even if you are considering a multi-center clinical trial which is planned in USA and you are going to apply it in Kerala, then obviously the Malayali version of the consent form is mandatory. Okay. That means local language version is mandatory. And what are the costs of multi-centric clinical trials? Yes, it will protect the research participant if the ethical approval is being reviewed by the local ethics committee. But the cons is that it adds significant delays to the study startup. Some ethics committee can give the ethical approval within 1 month. Some can take 6 months. Some can take 1 year based upon the timing of frequency of their ethical meetings. So multiple review may result in differences in the way patients are treated and at different sites, for which there is no ethical justification and may cause confusion among the participants. So there are some solutions which has been projected. So multiple IRB request different forms and systems for protocol submissions. But if we have a centralized ethics committee, yes, Indian Council of Medical Research has already established a centralized ethics committee at Bengaluru ICMR centers, and it has a defined centralized ethics committee format for submitting the protocols. So we can go through that also. So identify a standard data elements to facilitate review and reporting. Then we can have a regulatory reliability in the event of non-compliance of IRB. Means it can be a point when the separate ethical approval committee are giving separate ethical approvals. But if a central ethics committee is there, then it can clarify the regulatory policy to take action against the IRB or site for non-compliance also. So these are the points. Means you can have a centralized ethics committee. That can be a solution also. Coming to sample size, yes. The uncertainties and constraint for sample recruitment exist with each centers. It is not 100% assured that each study centers will have equal number of study sample size. We cannot expect that AIIMS New Delhi will recruit same sample size as that of a study center located at Northeast regions. So accordingly, we should allocate optimally the sample size to different study centers, which also should take into consideration that there should be minimal variance and it should increase the power of estimating the treatment effect. The sample allocation can consider based upon the following formula or statistical analysis formula. So total sample size, if it is n, then it can be the summation of different sample size allocated to different study centers. That is H1 to L, or if 1 to L number of study centers are there and each study center has n H sample size, the summation is n. So how to calculate NH? NH will be calculated by this formula, where NH is the total potential patients at the study centers and sigma H is the standard deviation of the endpoint at the study center H. Okay. Then divided by the root over of CH. CH means cost of obtaining data from each patient. Okay. So this will be divided by summation of H1 to L, NH into sigma H divided by root over of CH root over. Whole include into the total sample size to be required for the endpoint as per the multicentric study. So this is the formula to derive the sample size for each study center. So this will reduce the variance and increase the power of the study site. Otherwise, it will be very much difficult to enroll same study sample numbers in each study sites. So sample recruitment usually conducted in late phase of drug development, and rapid recruitment, that means thousands of patients recruited over 3 to 5 years of a period. And so that can be variation in the timing of recruitment. So even if you have allocated the study sample size to different study centers, but some centers can take years to collect the sample size. Some study centers will require only 1 or 2 months to require the samples to fill the sample size. So there will be variations. So the time period of recruitment is not a problem. Recruitment over decades would lead to will reverse phenomena like things. What is will reverse phenomena? We'll see it later on. And there can be changes in the institutional review boards, multiple layers of reviews, staffing changes, and some issues can come if the study will take decades to fulfill the sample size. So see this example of will reverse phenomena. So this is a cancer study, and this is prostate cancer study. And initially, this is the Gleason staging was being followed for the prostate cancer stagings. But during the course of the study, the Gleason's scoring system has been changed. So you can see this is original scoring, this is contemporary scorings. So based upon this, your sample size requirement varies. Okay, so the percentage of men which is having the different stage of the cancer as per the initial staging is different when we are considering the current staging, because this study has encompassed or encroached decades for this study sample collections. So coming to randomizations, yes, the coordinating center should perform these randomizations, and it should be a central randomizations. Ideally, it should be a stratified randomizations. And what should be the criteria for stratifications? The strata may include the study centers as a strata, or it can include different other parameters like the cost of enrolling the subjects, the type of the disease, or different endpoints can be considered for other different strata also. So the thing is that you should ensure that a site discontinuing participation in the trial or enrolling poorly would not affect the overall balance of the treatment groups. Coming to the power of the study and design effect, is the multicentric clinical trial design can be homogeneous and a balanced, or it can be heterogeneous and cluster distributions. The thing is that we cannot expect that all study centers will have similar infrastructures or similar efficacy or potency to recruit the subjects and similar power to analyze the data or collect the data. There can be some variations based upon the investigators, based upon the disease or observer variability. Different things can be employed, which can give rise to different variation in the data being enrolled or collected. So this is called design effect. So design effect, if it is less than 1, it is called homogeneous group distributions. Then the power is increased and need less sample size. But if the design effect is more than 1, it is called heterogeneous and there is loss of power and you need more sample size. So the design effect should be considered while considering the sample size. Otherwise, you will have a loss of power. That means you need more sample size if there is heterogeneous populations. So you have to consider the design effect in the study sample size calculations. So what is the design effect? Yes, the design effect is a statistic. It is calculated by this formula 1 plus S minus 1 into rho. So S is a statistic that quantifies the degree of heterogeneity of a group distribution among the study centers. So S is the variability among study center to centers, variation between AIIMS New Delhi to AMU, Aligarh universities. But rho is the center effect. It is the inter-class correlation coefficient, that is correlation between the observation from the same centers. Even if you are recruiting 100 subjects from AMU, what is the variation between them among these 100 subjects? That is inter-class correlation coefficient. So this design effect should be multiplied with the sample size formula to calculate the final sample size. Otherwise, you will have loss of power in the study. So I will not go through this. Because of this inter-observer variability, you have variation in the designs or inter-center variability. Okay. Okay. So how to do the sample size data analysis? So data analysis should include the effect size or the treatment center variation effect. So treatment effect size estimation to cover the center differences, but not treatment by center interactions. So heterogeneity of treatment effects means you have to interpret the care and you have to take certain different statistical method to calculate the data or analysis the data. So generally the mixed models are used, or I'll show the picture here. These are the different statistical methods applied for data analysis. One is random effect method, one is mental Hensel method, one is generalized estimating equation methods. The last one is generalized estimating equations, small sample correction methods. So if the small number of centers are there, like 5 to 10 centers are there, then random effect is applicable. We can apply this random effect analysis methods. But if the samples, the study numbers are more, like 50 to 100 study centers, then better to apply this G small sample correction methods. And this has been published in a recent study in 2020, and it has shown that this is the better method to be applied for this data analysis. And regarding this monitoring, yes, we should have a data coordinating centers. DCCC should be there, and it should have a member who should be qualified, and they should have adequate knowledge of the protocols, like all other studies. But the DSMB should be constituted depending upon the risk of the study interventions and the seriousness of the underlying disease being studied. And coming to disqualification, yes, it is not that we have enrolled 50 centers means all the 50 centers should be adhered to the study protocols. They may not adhere, or sometimes they are not able to even enroll single subject in the total duration of the study. Something can happen, or they may not be able to get the ethical approval. So sometimes the centers should be disqualified or withdrawn from the multicentric study. We cannot withhold the multicentric study because of some centers are not performing. Okay, so we have to devise some methods, and it should be mentioned in the protocol part. So what should be the disqualification criteria, and on basis of what criteria? So disqualified because of protocol violations and fraud, inability to recruit the patients, and this data should be handled on case-by-case basis. And either it can be intention to treat analysis or poor protocol analysis, and along with that, we have to perform the sensitivity analysis at the time of data analysis. And see one example that has been regarding the study, that is rescue studies, which is conducted in 2017. It is regarding high volume hemofiltrations in adult born patients with septic shock and acute kidney injury. So this study initially was planned to enroll 10 sites, 10 study sites, and total sample size is 120. But during the course of the study, one site opted out after screening, one excluded due to not enrolling in 24 months, and 3 were replaced due to staffing turnover and lack of dedicated PI. So of the 3 replacement sites, 1 opted out again. So at the end, 7 study sites were there. Out of this, 5 were the original study sites and 2 were lateral recruited. So this can happen in any studies also. And you can see this green color is depicting for this study, rescue study. This green color is depicting the originally planned protocol. Okay, we have planned that as this study should be completed within 2009 to 2013. But this study continued from 2009 to 2019. Okay, you can see the duration of the study, how the study duration can vary because of the variation in the centers. So accordingly, we have to apply certain rules that is the center should be disqualified or should be withdrawn from the study, something like that. Otherwise, it is not able to finish the study in time. Coming to regulatory aspect, the trial should comply to the local regulations. As I have mentioned in the recovery trials, how these local regulations can be applied. So it can adhere to all the regulations to all the countries where it is being applied. Okay. And accordingly, some procedures should be mentioned in the methodology or protocol part, that if some investigators are doing something wrong or there is some deviations, then there should be something that can be warning letters, disqualification of the investigators, application of integrity policy, or this can be applied as per the norms and policy of the regulators. One more thing is that suppose a study is being conducted in India, US, UK, or multiple countries, then multiple drug regulators need to inspect that study at different study sites. So suppose one study site is at AMU, Aligarh University. Then one time the US inspector will come, one time the UK inspectors will come, one time the Indian inspector will come. So this will give rise to variation in the timing and loss of resources. So there can be some policy that should be devised that, yes, there should be collaborative inspection programs. So collaborative GCP inspection initiative is already there, being applied in different countries. See one example, that MHRA and PMDA, that is Pharmaceutical Medical Device Agencies, they have collaborated among themselves for any medical device research. The MHRA and PMDA will have a collaborative inspections. So the collaboration to exchange information and collaborative inspection. So this can be applied also, collaborative GCP inspection initiatives. So coming to publication controversy, yes, it is one of the major important thing for all the investigators. Suppose I am an investigator in a multicentric trial, I will definitely want that my name should be there in the publications. Okay, so but sometimes what happens, out of this some group of investigators, some investigators should try to publish them in their own name only, and it has already happened. This is one of the example. Prophylactic granulocyte transfusions during chemotherapy of acute non-lymphocyte leukemia. This is the study being published by Drew J. Unson. Okay, and these are the investigators, but one of the four investigators, these are the four investigators, but out of them, one of the four investigators independently published the results without informing the sponsor and other investigators. So this is a ethical issue and also publication ethics issue. So for this handling, how to handle this? So there should be a prior publication committee should be established at the time of protocol development. So develop publication goals, guidelines, and timelines for proposal submissions. So the corresponding author specifies and registers the research group name and clearly identifies the group members who can take credit and responsibility for the work as an author. We can devise some formula that based upon the weightage, based upon the number of sample study subjects recruited, or based upon the data recruited. We can give some weightage based upon different criteria also to give the ranking of the authors also. So since the investigators from different sites have contributed, they qualify for the authorship. Means all the investigators from all the study sites are qualifying for the authorship, but there should be some ranking systems. So conclusions is global multicentric clinical trials identify and tries the challenges and demanding tasks with regard to the study planning, execution, analysis, and follow-up. Yes, we should have a digitally connected ecosystems and collaborative research networks, which will help in coordinating the different centers to overcome the common challenges in multicentric clinical trials. And successful completion of multicentric clinical trials needs a realistic timeline and prompt strategies, and frequent collaboration and frequent discussions among the stakeholders. It is not that only single time you have sent the protocol to the investigators and you will sit down as a project lead and other investigators will do their duty. No, you have to have in touch with all the investigators frequent basis. You should devise a time that every fortnightly or every 3 monthly we will have a meeting like this. So inevitable obstacles should be discussed in the pre-trial phase, and it should have a continuous correspondence. It is very important. It should have a continuous correspondence among all the stakeholders. And there should be maintenance of all relevant research parties throughout all the phases of the study in a same platform. You should not say that yes, this center is not performing, so we will not talk with him. We will not talk with that investigator. So we'll, either you have to follow the disqualification criteria, otherwise you have to take him on the board. You cannot leave him out. Okay. So this is the conclusion of a multicentric clinical trials and how to have a collaborative research network to have a fulfilled and good, robust clinical trials. Thank you. Any questions?
Thank you so much, sir, for such an informative presentation. We will now take up a couple of questions from the participant side, sir. So the first question that they have asked is that, are there any initial legal administrative agreements between different centers going for multicentric studies, sir? Over to you, sir.
Yeah. The thing is that as I have just given an example, during the trial protocol development, you have to take all the investigators in the board. And if it is a multicentric trials, you have to take consideration of all the different regulators also. And if it is a multicentric trial, you are the project lead, you have to get or you have to take the consent of or investigator declaration form and the endorsement form of all the institutes, and take all the certificates and compile them as one single PDF, and you can submit it. So the initial legal administrative requirement is needed to have a common platform and common form to be distributed to all of them and to get it signed and submitted to the project lead. Similar thing is that project lead should take account that all the investigational side have got their ethical approval also. Yeah. Who registered central registration of patient in
Thank you, sir.
multicentric clinical?
Now the last
There is
question
another question.
for you, sir.
Yeah. Yes, yes. Yeah, who registered central registration of patient in multicentric clinical trials? Yeah, central registration should be maintained at the coordinating center. Okay, means it, otherwise it will be very much difficult at any point of time. Suppose the regulatory is asking that what is the total number of patients recruited till now? Like in the recovery trial I have shown, 48,465 patients have been recruited till date. How you are getting this data? So there should be central registration systems. How many center is recruiting how many subjects? It should be immediately reported to a central registration desk, so that we have a compiled data that is till now how many total subjects have been recruited in these multicentric clinical trials. So this should be maintained at the coordinating centers. Yeah, any other?
Thank you so much, sir, for being with here today and answering all the questions. I extend my heartiest thanks to you again, sir. Thank you, sir.
Thank you.